I'd like to thank the CFSRE for the opportunity to speak at today's symposium, and it's my pleasure today to speak about the changes in the cannabinoid NPS landscape. Cayman Chemical is a proven leader in this cannabinoid NPS field, and we have continued to keep pace with the introduction of new reference standards and certified reference materials for both synthetic and semi-synthetic cannabinoids. To understand the current NPS cannabinoid field, it's important to reference the changes brought about by the 2018 Farm Bill, which removed scheduling on hemp and its derived products such as CBD, so long as hemp was defined as cannabis having less than 0.3 dry weight % delta-9 THC. This opened up a whole new market for psychoactive products like delta-8 THC to be sold in places where cannabis use was both legal and where it's still prohibited. This conversion of an isolated hemp product such as CBD into a psychoactive substance such as delta-8 THC is what defines a semi-synthetic cannabinoid. I think it's important to point out that while delta-8 can be isolated from cannabis, it's typically in very small, minor amounts. That's because there's not a biosynthetic pathway in cannabis to make delta-8. In reality, delta-8 THC is actually a degradant produced during storage or isolation. We've seen certain legislation that targets delta-8, with some places banning delta-8 products altogether. However, other semi-synthetic cannabinoids have emerged and are taking its place in these hemp-derived vapes and gummies. This is exemplified in this chart from a recent Journal of Forensic Science paper that shows the explosion of semi-synthetic cannabinoids just in eastern Denmark over a 5-year period. So why is there this interest in semi-synthetic cannabinoids? And it comes back to the main psychoactive constituent in cannabis being delta-9 THC. The conversion of CBD to THC was known as far back as the 1940s with work done in Roger Adams' group at the University of Illinois. But it wasn't until the 1960s that Raphael Mechoulam's group was able to identify and definitively assign the structure to delta-9 THC for that psychoactivity to be associated with cannabis. That provided a basis for work to define how CB1 binding affinity and psychoactivity were related to structure. So some of these semi-synthetic cannabinoids actually appeared in those literature reports from Roger Adams' group in the 40s, and that included the synthesis and psychoactivity for both delta-8 and the hexahydrocannabinols. After Mechoulam's confirmation of the THC structure in the 1960s is when we first start to see synthetic cannabinoids appearing, compounds like HU210 or the CP compounds, which were found to be potent CB1 agonists and maintain some of the structural aspects of delta-9 THC. John Huffman's group was able to expand on this with the indole structures that we now more closely associate with synthetic cannabinoids like JWH018. That brings us back to the present day, where we're starting to see a lot of similar trends to semi-synthetic cannabinoid space that we saw for those synthetic cannabinoids. And while these semi-synthetic cannabinoids are often described as hemp-derived, it's more, we have found more than enough evidence to see that some of these compounds aren't being isolated from hemp, but rather they're fully synthetic. And so you're seeing a merge and blending of these two synthetic and semi-synthetic spaces together. So structurally, you can define synthetic cannabinoids as possessing 3 distinct structural features. The core heterocycle, typically an indole or endosole piece, a headpiece that has many different structural functionalities, and finally an aliphatic tail. Likewise, the semi-synthetic cannabinoid scaffold can be thought of as modifications of delta-9 THC. Isomers are changes in the location of the double bond in the cyclohexene ring. Homologs are the addition or subtraction of methylenes in this typical 5-carbon alkyl chain. And finally, analogs are produced either by chemically modifying the olefin or by adding a functional group to this phenolic oxygen. Usually the addition of these protecting groups allows this to serve as a prodrug for the delivery of THC. Another trend we're seeing is the attractive packaging of these semi-synthetic cannabinoids in the vapes and the gummies that are appearing in. But we also are seeing reports of the semisynthetics being applied back to cannabis flower to end up boosting its activity. I'm going to explore some of the semisynthetic products and we describe as isomers, homologs, and analogs, and how these are kind of blending together to increase their complexity for analysis. So again, the CBD conversion to THC is known to make several isomers. The primary isomers being delta-9 THC, which under these acidic conditions readily isomerizes to delta-8 THC as the thermodynamic product. However, during this process, other isomers are also formed, including the exo-THC, where the double bond has now moved between carbon 9 and carbon 11. Additionally, these iso-THCs form because CBD has two olefins that the phenols can cyclize on. And when the phenol cyclizes into the cyclohexene ring, the iso-THCs are formed. It's important to point out that these iso-THCs are not found in cannabis. Therefore, they serve as powerful markers for that CBD conversion process. There's also compounds like delta-10 and delta-6A-10A THC. These isomers can be formed during isolation, but they're more directly formed via conversion from delta-9 THC. We have our sponsored workshop later today where my colleagues will be discussing these two semisynthetic compounds and some of the work we did to identify the potential metabolites. I hope you'll attend. Finally, delta-7 THC and delta-6A7 THC have also been described in the literature, but they have lengthy synthesis, mild to no activity, and they have yet to be identified in semisynthetic cannabinoid products. Moving on to homologs, it's well known that cannabis has some phytocannabinoids with varying chain lengths, with THCV being the most well-known, which has a 3-carbon alkyl chain. Starting in 2019, there were some reports of other homologs being identified in certain chemotypes of cannabis. These included the 4-carbon butyl, the 6-carbon hexyl, and the 7-carbon pherol. Each of these homologs can be identified by the addition of a letter to the end of the THC acronym. In the case of THCP, the activity for this at the CB1 binding affinity was calculated to be 30 times greater than that than compared to THC. So this really generated a lot of interest in these compounds. However, when you go looking for how much THCP is actually in the cannabis plant, you find that even in the most abundant chemotype studied in this Journal of Natural Products paper from 2021, it's only present at less than 0.02 dry weight %. So it's a very minor component, but we're still seeing THCP products appearing and marked as hemp-derived. And so is it possible that these are being extracted? And it's not likely. During this same time period, the availability of the chemicals to make the homologs has increased from both international and domestic suppliers, while the prices have significantly decreased. So while we still consider these semisynthetic cannabinoids based on their advertisement as hemp-derived, they're more likely fully synthetic, and that makes them subject to the side products generated during synthesis, such as this dicoupled CBDP or this iso-THCP. Finally, I want to talk more about analogs. I mentioned the earlier work by Adams' group that described the HHCs in the 1940s. And they are popular in the semisynthetic cannabinoid market because they are specifically not a THC. And so they get away from that stigma and the regulations surrounding that THC acronym. And their ability to retain some psychoactivity makes them popular in vapes. As you might expect, this same chemistry has been applied to homologs like THCP, with HHCP products being marketed as hemp-derived as well. Another analog of importance has been the THC acetates. They're often referred to as THCOs. The acyl group is attached to the phenolic oxygen, and it serves as a prodrug to deliver the parent THC during dosing. In the case of inhalation, it's been found that under vaping conditions, these compounds are thermally unstable and can produce ketene, which is a potent acylating reagent and can fall apart to acetic acid. Newer esters have been entering the market either due to legal issues or other safety implications. These include the butyrate and isovalerate esters and this new methyl carbonate analog that has been reported. And I'd like to highlight a new paper that analyzed products in Japan advertised as having CBP or THXE. The THXE acronym isn't associated with a readily identifiable product, but CBP is actually a phytocannabinoid acronym for the 7-carbon homologue of cannabinol. But the researchers did not find that in these oils and herbal products. Instead, they found a number of homologues and ester analogs, including THC-H acetate and both delta-8 and delta-9 THC propionate. So it's important to see that these three compounds have the same chemical formula, so they're isobaric, and that shows the importance of having a reference standard for being able to positively identify each compound. They also found THCP acetate, and they observed the acetate and the propionate for these iso-THCs, which shows that these came from a CBD conversion. So I've shown that the trends that defined the synthetic cannabinoid epidemic are now being repeated in this semi-synthetic cannabinoid space. This complexity appears to be increasing by the day as people are making the different isomers, homologues, and analogs and mixing them together. I think this recent post to a cannabis extraction board signifies some of the potential dangers. This user is advertising that they have a fully synthetic cannabinoid that has a delta-8 core, and instead of the alkyl chain, they have an adamantyl group. They describe the purity and even the potential impurities that are present, and they finish by saying that they can't help but be curious. I want to acknowledge the talented scientists I work with here at Cayman who make and characterize these reference standards. We always appreciate the customer feedback and hearing about what you're seeing in your own samples in order to provide you with the most reliable and up-to-date standards in any of the NPS fields. I hope you'll also join my colleagues this afternoon for the workshop on semi-synthetic cannabinoid metabolism. And I appreciate your time and attention and would be happy to take any questions.
Brilliant. Thank you so much, Jeffrey, for that really, really interesting presentation. And I'll just say, hopefully we'll get some questions coming through in the chat. But in the meantime, I was just wondering, how prevalent are these iso-THCs in semi-synthetic cannabinoids?
Well, it really depends on the reaction conditions that the chemist is using to make the CBD conversion to THC. Under some reaction conditions, they can be upwards of tens of percents of the overall THC content. But because they tend to coalute with both delta-8 and delta-9 THC, they tend to go undetected. So you have to use a specialized method to sometimes detect these. There are some HPLC methods reported for being able to separate them out, but they're very long. So typically a GC method is employed to be able to positively identify them.